Professor Jinho Kim of the Department of Biomedical Engineering. The title of his talk is Repairing Damaged Lung Tissue via Site-Specific Cell Replacement. Jinho, you're on.
Okay. Hello, everyone. This is Jinho, and thank you, Henry and Jean, for organizing such a wonderful seminar. So I'm very happy to present my work here today. Okay, so I would like to talk about the cell replacement therapy approach that my team, research team, has been pursuing to repair damaged lung tissue. Okay. So lung disease is one of the leading causes of global deaths and hospitalization. So more than 200 million people around the world are suffering from lung disease, such as lung infection, lung cancer, and pulmonary fibrosis, and also COPD. And unfortunately, nearly 100 million people die every year because of this lung disease. And because of the air pollution and increasing cigarette and e-cigarette smoking, and also emergence of new pathogen we never faced before, such as novel viruses and also drug resistance bacteria, more people are getting lung disease. Okay, so most of us when we get lung disease, we recover, but people who are end-stage lung disease with incurable end-stage incurable lung disease, such as COPD and pulmonary fibrosis, the only therapeutic option for them to prolong their life is to get a new pair of lung from organ donors. So this lung transplant procedure has been successfully performed in early 1980s in University of Toronto. Since then, more than 40,000 people were saved from this lung transplantation. And these days, nearly 2,500 procedure is being performed in United States. Notably, lung transplant has been used to treat the COVID-19 patient with severe lung injury. In early this month in Chicago, a young female patient received a new healthy lung from organ donor, and she was recovered from the disease. And as you can see here, her lung, due to the coronavirus, was completely damaged. And notably in China, it's been already 6 lung transplantation has been performed to treat this COVID-19 patient. So while this lung transplantation is only option for many end-stage lung disease patients, because there are not many lungs that can be transplanted, always there are more people who are waiting to receive new lungs than people who actually receive. While around 2,500 procedure has been performed, there are more than 3,000 people every year waiting to receive a new lung. Because of the lack of viable lungs, people have to wait more than sometimes 1 year, and in some cases 3 years. Although many people eventually receive the donated lung. While waiting, because they are very sick, they are end-stage lung disease patient, many of them die from the disease, and also some of them are removed from this organ transplant wait list because their symptoms get worse, and doctors think that new lungs will not even help them, so they are removed. And the problem of this organ transplantation is there are always a lot of people who are trying to receive these new organs. There are very few number of donors and the organs that are donated for the transplantation. So as you can see in this figure, every year the number of people looking for new organs are increasing very rapidly, while the actual donors and the transplantation is not increasing as rapid as the number of people. For the lung donation, one of the challenges is only 1 out of 5 donated lungs can be transplanted, because at the time the lungs are donated, they are severely damaged. So if you and I end up being organ or lung donors, only 20% of the lungs can be transplanted, while the majority of lung can be rejected for transplantation and thrown away. So this is such a waste. So our research team's goal has been to repair some of the refused donor lungs. So not all the lungs, because many lungs that are rejected for transplantation are severely damaged, but some of the lungs maybe we can repair by using a stem cell and biomaterial implantation technique. So the schematic of this procedure is briefly shown here. So the way we do is to obtain this the marginal lungs, so the questionable lungs that transplant surgeon may just reject for transplantation. We obtain those lungs and bring them to our research lab and remove the damaged cell that caused the lung rejection and implant the new healthy cell that are grown inside the lab. And in the bioreactor, we allow the new cell implanted to recover the injury in the lung. And then we can give the new lungs, bioengineered lung, to more recipients. But this approach is really difficult because the lung is really complex organ structurally and also biologically. But in many cases, the injuries at the epithelium layer, which is the outer surface of the airway tissue. So the way our team is doing is to first remove this damaged cell in the damaged area of the donor lung by introducing a small volume of the liquid that contained the enzyme or some chemical that can dissociate the cell membrane or detach the bindings of the cell onto the tissue structure. So by very gently introducing this liquid into this injured region, we can remove this damaged cell very carefully. And we demonstrated this approach using rat lungs. So this is the cross-section of the lung that we isolated from donor rat. And as you can see here, this is the airspace, and this is the epithelium layer that are detached by introduced the enzyme and also the chemical through this airspace. And most importantly, we want to just remove the damaged epithelium while preserving the normal tissue. And we evaluated the other tissue components such as cartilage, as you can see here in the blue color, and also blood vessel, the red membrane here. And also this procedure didn't damage or change the microstructure of the lung tissue, as you can see in the SCM images. So after we remove this damaged cell, what we have to do is to introduce a new healthy cell into this the compromised region. So we do we use a similar method to introduce this cell. So by forming very small plug of this cell suspended in a small volume of liquid, by introducing them through this airway, by applying positive pressure, the cell can be deposited onto the surface where the damaged cells are removed. So the cell can be attached and repair or recover the function of the tissue. So we also demonstrated this approach by using isolated rat lung. So this is the rat airway that we imaged using a fiber optic imaging probe we created here. And this video shows the stem cell that are introduced and attached onto the epithelium, onto the airway surface after the epitheliums were removed. And not only the airway, but also we were able to introduce this stem cell all the way into the gas exchanging the alveoli space. So this is the human lung. They were rejected, but donated for research. So we introduced the stem cell through the trachea of the lung. And we were able to see the cell labeled with a red quantum dot attached onto the surface of the alveolar surface. And this cross-section of the tissue showed also the cell that we introduced were nicely attached onto the alveolar surface. So this cell replacement treatment can take many hours and sometimes a couple of days. So we really have to provide the lung because they are removed from the body of the person. We have to really provide the physiologically relevant condition to the lung during this intervention. So we created this lung bioreactor system by integrating clinical ventilator and a perfusion pump that perfused blood. The pump is not shown here, but the pump can perfuse the blood or blood-like solution to the lungs so that the lung can feel like it's inside the human body. In addition to this ventilator and the perfusion pump, we created and incorporated some imaging systems such as portable X-ray imaging system that can allow us to monitor the recovery of the lung, and also customized fluorescent microscope that allow us to see the cell delivered into the lung. And also the bronchoscope that we used to inspect the airway surface inside the lung. And also we use a thermal camera to monitor the temperature change of the lung to understand the perfusion of the blood throughout the lung. So using this, some technique that we developed, we were able to repair or recover the function of the lung initially damaged. So this is the pig lung. We created the injury by introducing the gastric contents. So many donors, lungs are damaged because while they are lying in the bed, the fluid from their stomach entered their lung. So this lung was damaged by introduced this gastric contents. And as you can see, some part of the lung was collapsed, which means they are not participating in the ventilation or the gas exchange. And over time, the color of the lung got more red because the blood is being perfused throughout the lung. And also the majority of the region of the lung that was initially collapsed was recruited. And also the thermal imaging shows that initially, because the lung was damaged, lung is cold while we are introducing this warm blood to the lung. But over time, the lung, the blood vessel was also repaired, and the blood circulation was more uniform throughout the lung. So the temperature become more like the temperature of the blood that we introduced. And also the histology showed that initially the damaged alveoli septum, this tissue is called septum, is inflamed because they're injured and damaged, but over time they become thinner, so the gas exchange can occur much better, and the more airspace we were able to have. So that's all I wanted to present, but with this, I would like to thank my collaborator that I have been working with in the past years, and also the funding support, including the Stevens Bridging program that allowed me to build a system to do some of this experiment. And also I would like to thank my PhD students, especially Mohammed and Jaman. They are working really hard, and also some other undergrad and master's student who has been working with me since I joined Stevens. Okay, so thank you very much for your attention. And if you have any question, I would like to answer.
Thanks very much, Jinho. Fascinating work. I have a question here. And your work is focused mostly on, or mainly on, repairing damaged donor lungs, right?
Yeah.
Is the approach applicable for repairing damaged lungs in living person?
Yeah, so that's a very good question. And I think that's the $1 million question. So our lab is also doing that, looking into that the possibility. So essentially, the COVID-19 patient, the reason why they are dying is because their lung is injured, and the injury mechanism and the consequence of the injury is very similar to this some of the lungs that are rejected, which is the ARDS, or acute respiratory distress syndrome. So we are looking into the possibility to use our method to repair the patient, live patient with severe lung injury.
Yeah, indeed. If you could find a good solution, we're talking about multi-million dollar solutions, right? That's great. Excellent work. Thanks much.